Vitamin B5, also known as pantothenic acid, is a B vitamin, therefore it is used in some of your cellular processes to release energy. It's water-soluble as a B vitamin, therefore toxicity is very rare because any excess can be excreted by the kidneys. As well as this, deficiency is also very rare. That is because pantothenic acid can basically be found in every single food. It's also in the name. Pantothenate is in the ion of pantothenic acid. Panto means all, and thenate means strength. So it's literally found in every single food. And it's not like a vitamin where the reason its deficiency is rare is because it's not really needed that much. It's actually a really useful molecule that's useful and is used to make a molecule that you probably would have heard of before. And that is a molecule called coenzyme A. And coenzyme A is used to make things such as acetyl coenzyme A, which is an even more useful molecule than CoA itself. This is why that it's found in most foods. Nature is kind of cool and it all kind of works together in a little interdependent cycle. Pantothenic acid is found in every food, and that is because CoA as a biological molecule is so vital for the human body. Without it, you couldn't have acetyl-CoA, and without acetyl-CoA, you essentially couldn't exist. That's why you can't name a deficiency. There's not even a name for the deficiency of vitamin B5, like there is with vitamin B3, with pellagra or something. It's unnameable because you can't isolate any symptoms, and it's so rare anyway because nature works in such a cool way. And as I've mentioned before, the main use of coenzyme A is in acetyl-coenzyme A. Coenzyme A is one of the cofactors in the big enzyme pyruvate dehydrogenase, where you're going to have pyruvate turning into acetyl-CoA, and a whole load of stuff happens in the middle of that. There's a lot of cofactors that go into it. You've got a whole big enzyme complex, and coenzyme A will be added right at the end of this sort of sequence, and I'll go through the mechanism of that now. If you watch my video on vitamin B1, thiamine, you'll also know that that is a cofactor inside of the pyruvate dehydrogenase enzyme complex, and this forms a complex called hydroxyethyl TTP, which is thiamine pyrophosphate, the active form of thiamine, combined with the pyruvate molecule after it's had some carbon dioxide excreted off of it. Now, hydroxyethyl TTP, that's not acetyl-CoA, is it? And so what's going to happen is a lipoamide that is enzyme bound, it's got a little sulfur ring, like it's going to have a disulfide bond, and then it has a little like 5-membered or 3 other carbons in a ring essentially. That is going to steal the acetyl group off of the hydroxyethyl TTP, and that's going to make an acetyl lipoamide. Now, a coenzyme A molecule is going to come along, and it's going to do a lot of like substituting things, like it's going to attack this, which is going to attack this, a lot of arrows and curly arrows and stuff. But eventually, you're going to have the S on the coenzyme A molecule, the little thiol group right at the end, which did have a hydrogen connected to it, but now that hydrogen has gone off and gone and bound to something else, and it's going to steal that acetyl group to form acetyl-CoA, which is great because now that can go into the TCA cycle, that can use to make ATP, which is literally how your body is used to make energy. But as well as the obvious, the TCA cycle, making ATP and stuff like that, you're also going to have some acetylation reactions where you're going to have an acetyl-CoA molecule combining with another molecule to make acetyl plus another molecule. A good example of this would be acetylcholine. Acetyl-CoA plus choline will make acetylcholine plus CoA. That CoA is then completely able to just go back into loads of other things and do some more uses. Acetyl-CoA is basically the main use of coenzyme A existing. It's kind of a useless molecule otherwise. I've only seen one other use of coenzyme A that's not literally making acetyl-CoA, and even then it still has something to do with acetyl-CoA, and I'll get onto that later. Now, acetylcholine is an interesting molecule. It's a neurotransmitter, and if you think of how a muscle works, how a muscle contracts, acetylcholine is responsible for your muscles being able to contract. A synapse where it's got a nerve ending and then a muscle sort of receptor. Acetylcholine is going to be a neurotransmitter that when the electrical impulse comes along, the acetylcholine neurotransmitters are going to be released onto the muscle. There's loads of receptors there, and the muscle is stimulated enough to be able to contract. That's literally how it works. It seems so complicated, but if you just flex your bicep, that's what's happening. Acetylcholine is being released in synapses as you do that. Acetyl-CoA is also a useful molecule because it starts cholesterol synthesis, which was probably the first sciencey based thing I ever made a video on, and it starts with two acetyl-CoA molecules combining to form acetyl-CoA, which also then combines with another additional acetyl-CoA molecule to form like HMG-CoA, which then eventually you get cholesterol. God knows how. If you want to see how, watch that video basically. But now the other singular use of coenzyme A actually is in beta oxidation and fatty acid synthesis, which still has something to do with acetyl-CoA. You have a long fatty acid chain, right? And you put it with a coenzyme A. You combine the two together, and that's going to mean that when you do all the reactions on the actual end of the fatty acid chain, it's stabilized by the coenzyme A, and eventually you chop it and change it so that all of the little 2 carbon segments on that fatty acid have now been made into acetyl-CoA, and that requires a lot of CoA molecules. And then you're also going to have the opposite of that, which is fatty acid synthesis, which is just that in the reverse direction. It might be a little bit different, it might not be exactly the reverse of beta oxidation, but it's got a similar sort of chemical premise. Now, pantothenic acid, as well as being basically existing in every single food that we eat, it also can be made in very negligible amounts by gut bacteria, and that can then potentially be absorbed in the gut, but it also could just stay in the bacteria. But I think that in some cells, you do have the ability to make pantothenic acid, and you make pantothenic acid from the amino acid beta-alanine and pantoic acid. Pantoic acid is an interesting molecule to make. It essentially starts with you combining 2 pyruvate, and then you build up from there, make little teeny tiny adjustments and stuff. I'll get like a graph or diagram chart to show you how the thing actually moves along. It is pretty cool, and it's also cool how when I've done biochemical research, a lot of molecules literally just start off from pyruvate combining, acetyl-CoA combining, glucose having this convert to that, and all of the base molecules, the proteins and amino acids, can make loads and loads of different things. It's really cool. But it's cool how simple pantothenic acid can be derived from 2 pyruvate and then a few little adjustments, as well as a beta-alanine amino acid, which is so simple when you think about it, but pantothenic acid, when you look at its structure, seems so alien to anything you may have seen biologically before. But with that, that's about all I have to say about pantothenic acid. It's a very incredibly useful vitamin, but it's very hard to be deficient in it. But don't let that make you get complacent, because there are other vitamins that exist which you do need to worry about. But with that, I recommend subscribing for more content like this, and have a good day.